right, everybody. Today we are going to talk about DNA. This is literally one of my favorite topics. I love DNA. It is deoxyribonucleic acid, and it is amazing. It is the genetic material that runs our bodies. And to start with, we are going to talk about the requirements for the genetic material. So what does it mean to be genetic material? Well, you have to be able to store genetic information. I mean, that seems obvious. You must be stable and able to be replicated accurately during cell division and then transmitted on from generation to generation. You must be able to undergo mutations in order to provide genetic variability. DNA contains two nucleotides with purine bases, adenine A and guanine G, and two nucleotides with pyrimidine bases, thymine T and cytosine C. So when we are talking about DNA, we are talking about A, G, T, and C. Only those four nucleotides can be involved in DNA. And according to Chargaff, basically the amounts of A, T, G, and C in DNA are constant among members of the same species and vary from species to species. Okay? All humans have the same amount of DNA, but humans and dogs do not have the same amount. Yet all dogs have the same amount of DNA to other dogs. Okay? In each species, there are equal amounts of A and T and C and G. And this is because of what we call base pairing. And A can only pair with T and G can only pair with C. So that is what we call complementary base pairing. Each human chromosome contains on an average about 140 million base pairs, leaving the number of possible nucleotide sequences to a very large number. Okay? Lots of possible combinations. How did we figure this out, right? How did we figure out DNA? Well, let me tell you. Rosalind Franklin, okay? She was studying X-ray diffraction, and she studied the structure of DNA using X-rays and this diffraction method. She found that if concentrated, viscous solution of DNA, it can be separated then into fibers. Okay, if you concentrate these solutions down, they can then be separated into fibers, and under the right conditions, she was able to produce an X-ray diffraction pattern. And through that pattern, through those photographs that she produced, she was able to see that DNA is a helix, and that some portions of that helix are in fact repeated. Now, you may say, well, I have never heard of Rosalind Franklin. And to that I say, well, that's a shame, because I didn't know about her right away either. I feel like when I learned about DNA, we learned about Watson and Crick, right? The two guys that won the Nobel Prize for coming up with the structure of DNA. But in reality, it was Rosalind Franklin who took these photos, and a friend of hers then showed this photo to James Watson, and he said, I do know what this means. Now I see the structure of DNA. So it was really a joint effort. And Watson and Crick could not have come up with their theory if they did not have that picture from Miss Franklin, right? Okay, having said that, in 1953, they come up with this Watson-Crick model, which is a double helix, which is basically a twisted ladder, and that is the shape of DNA. Okay, so the backbones of the ladder are the sugar phosphate, sugar phosphate, sugar phosphate, and then there are hydrogen bonded bases that are making up the rungs. So if you are thinking about a ladder, the part your feet are stepping on are the rungs. This is the nucleotide bases, and the part your hands are hanging on to the sides are the sugar phosphate backbones. Okay? And you have two DNA strands that are running anti-parallel, meaning one side is going in one direction and the other side of those handles that you'd be holding on to is coming down the other way, meaning there is a specific direction that DNA is read in, and that is, here it says, information stored in DNA must be read in the 5-3 direction, because that is how DNA is going to be replicated. It is only replicated in this 5-3 direction, and that has to do with how the bases are connected to the backbone, and the backbone is connected, the sugar and the phosphates are connected to one another. Okay, so complementary base pairing ensures that one purine is always bonded to one pyrimidine, meaning A goes with T, G goes with C. And it was this knowledge that won them the Nobel Prize in 1962. And that is why we always hear about Watson and Crick. So, here we have this DNA. Well, what are we going to do with DNA? You know, what's the purpose of it? Well, it's all our genetic material, and we need to keep copying it because we are constantly making new cells, right? So in order to copy it, we have to, be able to replicate it correctly. So we are going to talk about the replication of DNA. And this is the process of copying a DNA molecule. DNA replicates semi-conservatively, meaning each strand of the original double helix serves as a template for a new strand in a daughter molecule. So what we're going to see is that ladder is going to open in the middle where your feet would go, and each of those strands will make a new strand, meaning every time you make a DNA molecule, you've basically got one old strand forming a new strand, which is why we say it's semi-conservative. You've got an oldie and a newie coming together to make a new DNA molecule. Does that make sense? So you have one of the parental molecules coming together and forming with a daughter strand. So, in order for replication to happen, a couple of steps must be followed. First, we have the unwinding or separation of the two strands of the parental DNA molecule. Okay, like I said, those rungs are going to break and it's going to open. And that is able to happen through the help of DNA helicase, which is an enzyme that comes in and cuts those nucleotide bases apart, opening up that strand. The single-stranded binding proteins, the SSBs, then attach to newly separated DNA and prevent the helix from coming back together. Complementary base pairing between a new nucleotide and a nucleotide on the template strand then starts to happen, meaning a DNA primase places short primers on the strands that need to be replicated. Polymerase recognizes RNA and begins what is called DNA synthesis. The two strands are replicated differently. There's what's called a leading and a lagging strand, and the joining of nucleotides in the lagging strand by the DNA ligase form the new strand. And then, as I said, each daughter DNA molecule contains one old and one new strand. In prokaryotic replication, we see a little bit different of a series because we know the prokaryote cells are not as intricate as a eukaryotic cell, right? So here we just have bacteria that have a single circular loop of DNA, and the replication moves around the circular DNA molecule in both directions, and therefore produces two identical circles. This process begins at the origin of replication. Replication takes about 40 minutes, but the cell divides every 20 minutes. So a new round of replication can begin before the previous round is even completed. So it's a pretty quick, somewhat basic process that we see in prokaryotic replication. In eukaryotic replication, it's a little more tricky. DNA replication begins at numerous points along each linear chromosome. The DNA unwinds and unzips into two strands, as we said. Each old strand of DNA is serving as a template for that new strand. And then through complementary base pairing, we see a new strand being paired with the old strand. And that means when those open, if you have an A sticking out, a T is going to come in. If you have a G sticking out, a C is hopefully going to come in, right? Because we know that A and T bind together and G and C bind together. So each open end on either one of those strands is hopefully attracting the correct paired nucleotide, ATGC. And this requires a certain enzyme called DNA polymerase because it is forming more of the chain. So, replication bubbles spread bidirectionality within a form of bidirectionality until they meet. So there's lots of different spots on the DNA that are being replicated at the same time. The complementary nucleotides are joined to form new strands, and this is why we call it semi-conservative, because we know that the old strand is going to make a new strand. So each daughter molecule contains one parental strand and one newly synthesized strand. Then we have the polymerase, which I mentioned, which is an enzyme that is unable to replicate the ends of the chromosomes, or what we call the telomeres, and they must be copied by a certain enzyme called enzyme telomerase. And if that is unregulated, we can see some problems usually in cell function. But we don't usually see that. So for the most part, our DNA is replicated pretty accurately. I mean, it's amazing. So the DNA polymerase is very accurate, yet it can make a mistake. Usually the average is about once per 100,000 base pairs. But it is able to first identify and then correct those errors. So usually, even if a mistake is made, the DNA polymerase will identify it and fix it. But if it is not caught and not corrected, it could lead to a mutation. And mutations are a whole nother lecture. So we are going to end there on our replication of DNA. I know it's a lot to take in. You may need to listen to this lecture twice if you have never heard about replication of DNA, because it is probably a little overwhelming at first. But once you read about it and you listen, it does make sense. And it's always the same process. There are no tricks. So we just need to make sure that we do understand the process. If you have any questions or concerns, please reach out. Have a fabulous day.